epistasis and modifier genes. Epistasis is a phenomenon that consists of the effect of one gene is dependent by the presence of other genes. The other genes, they are also called as the modifier genes. Epistasis is also such a phenomena in which alleles at one locus can prevent or hide the expression of the alleles which are present on another locus. Such a phenomena is called as epistasis. Epistasis can be of different types. One of the type is positive epistasis. In case of positive epistasis, due to the presence of double mutation, the phenotype becomes the fitter. In such a cases, there are some improvements in the functions of the organisms. While in case of negative epistasis, that is also due to the presence of two mutations. Due to the presence of two mutations, the severe effect or the deleterious effect becomes less or it minimizes as compared to the mutations which are present in independent genes. Sign epistasis. Sign epistasis is such a epistasis in which presence of mutation in one gene can revert or it can modify the effect of the second mutation. Reciprocal epistasis is such epistasis it occurs when two deleterious genes they are beneficial for certain traits. Epistasis between the genes can occur because there are different genes within there are different genomes. Within the genomes there are genes, and if there is epistasis between the genes, in such kind of the epistasis, it can be the direct epistasis and it can be the indirect. In case of indirect is epistasis in those genes that produce certain proteins. While in case of indirect epistasis is such a epistasis where genes they encode the components of certain cycles which are present or the metabolic pathways where those genes they express or there is a function of certain enzymes. Epistasis within the genes. Within the genes, epistasis can compensate the effect of each other. If there are two mutations within a gene, one mutation can compensate the effect of the other mutation. Such a mutation is called as epistasis within the genes. So epistasis is a phenomena in which the effect of one gene is modified by the presence of alleles at another gene locus. There are different types of the epistasis.